PathoGenetix has developed a revolutionary genomic analysis technology called Genome Sequence Scanning, or GSS for short. This technology is able to scan single DNA molecules, creating distinct genomic fingerprints, even when multiple targets and background are present in the same sample. The first commercial system which utilizes GSS is the Resolution Microbial Genotyping System. The Resolution system has been developed to provide molecular serotype and strain type identification of bacteria, even when present in mixed cultures. The Resolution system will process up to 8 samples at a time, providing results sequentially, with the first result available in approximately 5 hours, and the 8th and final result available after 8 hours. The first Resolution assays have been developed for the food safety and public health markets. Assays for additional markets, such as hospital infection control, industrial microbiology, microbiome analysis, probiotics and starter culture quality control, food spoilage, and biodefense are in development. The Resolution system is fully automated, providing molecular serotype and strain type identification of target bacteria in as few as 5 hours. The Resolution assay protocols have been developed for the rapid confirmation and identification of target pathogens in samples producing a presumptive positive result with a screening assay. This is an illustration of how samples might be processed for pathogen screening using the DuPont BAC system, followed by confirmation identification with the Resolution system. The technician is processing ground beef samples for salmonella screening with the BAC system. Pathogen screening of food samples is relatively straightforward. The technician weighs out 25 grams of the food, adds that sample to a stomacher bag, then adds 225 milliliters of the prescribed enrichment broth and thoroughly mixes the sample. Samples are then incubated for 18 to 24 hours at the designated temperature. After the enrichment period, 5 microliters is removed from each sample, added to a tube containing BAC's lysis buffer, and incubated for the prescribed time. After the lysis period, 50 microliters of each lysed sample is added to a tube containing the BAC's PCR reagents. Tubes are capped, and the tubes are then placed in the BAC's instrument for amplification and analysis. Samples generating a negative result are negative for the target pathogen. Samples producing a positive screening test result are considered to be presumptive positive and require further analysis for confirmation that the pathogen is truly present, as well as the identification of serotype or strain type. The Resolution system provides results days faster than other identification technologies without question. On day 1, samples typically go through a primary enrichment for 18 to 24 hours. On day 2, the enriched sample is then tested with a rapid screening method, providing results in 30 minutes to 8 hours, depending on the method. Presumptive positive samples could then be analyzed on the Resolution system, providing rapid confirmation, molecular serotype and strain type identification in as few as 5 hours for the first sample and 8 hours for an entire batch of 8 samples. All other identification and typing technologies require a pure culture isolate, which can take 3 to 5 days to obtain. Many of the molecular methods of identification take about 8 hours to generate results after the pure culture isolate is available, which correlates to day 6 on the timeline. Other methods, such as traditional serotyping, whole genome sequencing, and pulse field gel electrophoresis, are highly manual, require specific technical or bioinformatics expertise, and can take up to 9 days according to FDA protocols. Genome Sequence Scanning, or GSS for short, is the key technology that enables the determination of molecular serotype and strain type of target pathogens in a complex, mixed culture. Genome sequence scanning works in the following way. The enrichment culture that produced the presumptive positive screening test is loaded onto the Resolution system. The Resolution robot and fluidics adds the appropriate reagents and buffers at appropriate times in the GSS process. The bacterial cells in the sample are lysed using a combination of enzymatic and chemical processes. The intact genomic DNA is then cut into large fragments by a restriction enzyme. The DNA fragments are then labeled with 2 different protein nucleic acid (PNA) probes, each having a different fluorescent label. These probes are 8 nucleotides long, which results in each probe binding repeatedly throughout the genome. The complementary sequence for each probe occurs approximately once every 10,000 base pairs. The system also labels the DNA fragments with a nonspecific intercalation dye. The restricted and labeled DNA is then automatically transferred to the genome scanner, where the sample will be scanned and analyzed. The genome scanner utilizes a proprietary microfluidic chip, lasers, and optics to scan the DNA. The microfluidic chip has a dual funnel design, which forces the DNA fragments to unravel and take on a linear conformation. As the labeled DNA goes through the microfluidic chip, it is scanned by 3 lasers, which determine the length of the DNA fragment, as well as the spatial arrangement of the hybridized PNA probes. Single DNA molecules are scanned at a rate of 10 million base pairs per second. This single molecule scanning is what enables GSS to determine the strain types of target pathogens present in a mixed culture. Each scanned piece of DNA essentially generates 2 barcodes, one for each probe color. All of the barcodes from a specific strain type comprise that strain's GSS fingerprint. Onboard software and analytics automatically compare barcodes to the resolution database, which is comprised of fingerprints generated by well-characterized target strains and related bacteria. Results are automatically generated and the strain type ID is displayed, as well as a list of related strains and the level of similarity of GSS fingerprints. The resolution system protocols have been developed with simplicity and speed in mind. Minimal laboratory and technical expertise is required to run samples on the resolution system. The resolution system uses the same enrichment broth that produced the presumptive positive result, saving time and labor. A pure culture isolate is not required. The sample handling requirements for resolution analysis are simple and typically take 20 to 30 minutes for a batch of 8 samples. 2 milliliters from each sample is loaded into a microcentrifuge tube. The tubes are then centrifuged at low speed for 5 minutes. This removes any food material that is present. After the 5-minute spin, the supernatant is removed and transferred to a clean microcentrifuge tube. These tubes are then spun for 2.5 minutes at high speed. This concentrates the bacteria in the sample into a pellet at the bottom of the tube. After the second spin, the supernatant is removed and the pellet is resuspended in saline solution. Cell concentration is adjusted by taking a simple OD reading, entering the OD value in a spreadsheet, and adding the corresponding volume of saline. Sample tubes are then placed in the instrument. After loading the sample tubes, the user will unpack and load the open well reactor, or OWR, which is a one-time use consumable. The open well reactor is a proprietary consumable that enables the gentle extraction and processing of DNA, preserving the integrity and quality of very high molecular weight DNA. The sample is transferred from the sample tube to the corresponding open well reactor in the strip of 8 OWRs via the Resolution's liquid handling robotics. The sample fluid is drawn through the membrane at the bottom of the OWR, concentrating the sample on the membrane surface. The sample is essentially pinned to the membrane by a constant flow of fluid through the membrane. Reagents for lysis and protein digestion are sequentially added to the OWR, and cellular debris that does not pass through the membrane is eliminated when the system allows for the flow of solution through lateral ports and valves. The Resolution's sample processor allows for variable times and temperatures, providing flexibility in sample preparation protocols, and the OWR design allows for the sequential addition of reagents and wash buffers without having to physically move the sample, thereby preserving the physical integrity and concentration of the DNA. After the OWR is loaded, the user then loads the reagent strips. Once the reagent strips are loaded, the user then enters the sample information via the Resolution touchscreen. Once the user completes the sample info entry, the user simply presses Start, and all remaining Resolution processes, data analytics, and results reporting are performed automatically, requiring no additional user intervention. The following is an illustration of the different states or processes that occur during the sample prep process in the Resolution system. The Resolution's liquid handling system begins the sample processing by priming the 8 probes or needles. After priming is completed, the system proceeds to the wash state, in which the OWRs are hydrated and washed with buffer. After the OWR membranes have been wetted, the robot moves and draws up the required amount of sample and then injects the sample into the OWR. The system then enters the incubation state, during which time reagents are added, incubated, and washed away as the specific assay requires. Once all of the necessary incubations and washes are completed, the system will enter the elution state. At this point, the flow in the OWR is reversed, allowing the restricted and labeled DNA to move off of the membrane and be drawn up in the fluid probes. The robot then transfers the eluted DNA to clean tubes loaded in the detector turntable. After the eluted samples have been transferred to the detector turntable, the liquid handling robot proceeds with a decontamination and purge process, disinfecting the liquid handling components and preparing for the next batch of 8 samples. The second batch of samples can be loaded approximately 5 hours after the first batch is started. Once the eluted DNA is loaded on the turntable, the liquid handling robot in the genome scanner will load the samples one at a time to be run through the proprietary microfluidic chip. The restricted and tagged DNA is mixed with the intercalation dye POPRO as the DNA moves through the microfluidic chip. The liquid handling robot in the genome scanner operates in a similar manner as the robot in the sample processor and is illustrated in the following. The buffer reservoir is first filled with TE buffer. After this, the wash sipper and flush sipper pumps and lines are filled with TE buffer. The POPRO intercalation dye is then loaded onto the chip, which is followed by a flushing of the waste lines. After this step, the system will load the DNA sample into the microfluidic chip. The DNA will be mixed with the POPRO dye as it moves through the microfluidic chip, and the sample will be scanned by the detector as it exits the funnel of the chip. After the sample scanning is completed, the system will backflush and purge the microfluidic chip in preparation for the next sample to be scanned. Once the sample enters the funnel of the microfluidic chip, the DNA becomes linearized and is scanned by the laser optics systems, determining the length of the DNA molecule and the spatial arrangement of the bound probes, which show up as fluorescent peaks, creating a barcode for that piece of DNA. The onboard data analysis software will average similar barcodes and then compare the barcode to a database of GSS fingerprints created from pure culture isolates or whole genome sequence. The fingerprint of a bacterial strain is comprised of all of the GSS barcodes associated with that strain. The key to the resolution system's ability to identify individual strain types or molecular serotypes in a mixture of bacteria is the system's ability to scan single DNA molecules. The video in the top left shows fluorescently labeled DNA moving into the microfluidic funnel. The system automatically adjusts the flow rate to achieve the optimal concentration of DNA entering the funnel. The video in the lower left shows single DNA molecules in linear form as they exit the microfluidic funnel. The illustration on the right shows the multiple barcodes created for a strain. The combination of all of the barcodes represents the GSS fingerprint for that particular strain. The onboard data analysis software uses proprietary algorithms to compare barcodes generated by a sample to fingerprints in the resolution database and determining the level of relatedness of the strains. The system is able to report the presence of species, molecular serotype, and strain type of the target pathogen in the sample, and can even identify multiple strain types or serotypes that are present in the same sample, which is shown here by the data produced when 3 different Salmonella serotypes were present in the same sample. This is a phylogenetic tree for Salmonella based on the GSS fingerprints. Each serotype forms its own branch on this tree, and individual strains form additional branches within its specific serotype branch, demonstrating the resolution system's ability to discriminate based on molecular serotype and strain type. Results are automatically reported by the resolution software and can be viewed on the system's touchscreen or downloaded to a remote workstation. The results report can be configured to provide as little or as much information as needed, ranging from a simple yes/no the target organism was present, to the molecular serotype and strain type present with a list of the most closely related strains and level of similarity to those strains in the resolution database. The Resolution Microbial Genotyping System is the first commercially available product based on the revolutionary single DNA molecule scanning capability of Genome Sequence Scanning. The first assays available for resolution will focus on the rapid confirmation and identification of foodborne pathogens. The speed and simplicity of the resolution system will enable considerably faster responses to presumptive positive results in food production, enable considerably shorter times to identify the occurrence of a foodborne outbreak, and considerably reduce the time it takes to identify the source of the outbreak, ultimately saving time, money, and lives.